They're, as you all probably know, they're just getting ready to launch. And well, be honest with you, the bottom line is we're trying to assess, are they ready, do they have enough knowledge, are they ready to move forward without floundering too badly? We want them to flounder a little bit, but not too badly. So it's an idea for constructive review, constructive criticism. If they're off target by miles, we need to let them know that. And if they're on target specifically and precisely, that we let them know that too. So it's a time for any responses you may hear or get in written feedback are not meant to be, damning. They're meant to be helpful. So take them and grow with them. And any questions for me to start how we run this? You want to add anything?
We are recording right now for our Librarian Award. This is the inaugural year that the University Libraries is sponsoring an award, and you all are the very first group to present, so.
And just so everyone knows who's in the room, let's have the folks around the table introduce ourselves, and then hopefully the team will introduce yourself when you get started. So why don't we start right here?
Sure. Joe Love, Department of Natural Resources and Fishery Biologist.
All right, I'm Frank Cole, Director of the Gemstone Program.
Kristen Scandal, Associate Director of the Gemstone Program.
Tom Miller from Chesapeake Biological Lab and also their mentor.
Alex Carroll, Agriculture and Natural Resources Librarian and one of the team librarians.
Eileen Harrington, Health and Life Sciences Librarian, and the other team librarian.
Josh Newhart, I'm a fish biologist with the U.S. Fish and Wildlife Service.
All right, excellent. So you guys have lots of good eyes and ears paying attention, so impress us.
Okay. Good morning. My name is Lauren Emrine, and myself, Yasmin Antati, and Skylar Golt will be representing Team Savior today. We have 11 members on our team, and we'll say our names real quick.
I'm Yvette Mann.
I'm Natalie Watts. I'm Isha Agarwal.
I'm Trevor Mills.
I'm Yasmin Toddy.
Skylar Gold.
Zeke Gonzalez, Greg Matthews,
Bobby Fitzgerald, and Brian King.
Our project is about snakehead attack, the investigation, and organic reactions. Our agenda today, I'm going to be talking about our research problem, and then I will go into our literature review, which delves into past control methods of invasive species and past control methods of the snakehead. Then I will go into our research questions, and from there, our methodology. Skylar will be talking about our survey, and Yasmin will be talking about our genetic analysis. Okay, so our research problem, in 2002, the northern snakehead was introduced into the Potomac River system. It is native to East Asia, and since then it has grown and naturalized in the Potomac River system. It is both an ecological and economic threat in the system because it is an apex predator and it'll eat both bait fish and lower tier preying fish. And it also competes with the largemouth bass for food and for habitat. And if it gets big enough, it may be able to start to prey on a largemouth bass. It is also an economic problem because it can interrupt other valuable fish populations that anglers are fishing for, and it can also reduce the value of the other fish in the river. Past control methods of invasive species. Electrofishing is a popular method used to control fish. In the 1980s, the rainbow trout was introduced in Tennessee, and they used electrofishing to control the population density. This was tried on snakehead here, but because of the vastness of the Potomac system and the population density of the fish, it's not feasible, and it also affects the other fish in a negative way. There is also chemical dumping that can be used to control the fish populations. In the Great Lakes, the sea lamprey, which is a eel-like fish species, was introduced to that area. They had found chemicals that were aimed directly at that fish, and every year they dumped this chemical into the water, and it effectively controlled that population. In Crofton, when the snakehead was first discovered, there were chemicals dumped into that lake, and it effectively killed pretty much every species in that lake. So that's, unless we had our 3 years to spend finding a chemical to target the snakehead, we wouldn't be able, that's not feasible in our 3 years, and we decided to move on to different social control methods. So state laws here in Maryland, if you catch a snakehead, you have to kill it immediately. and you cannot sell it commercially. In New York, and Virginia, you can also not sell the snakehead commercially. For other social controls, the Maryland Department of Natural Resources holds a competition where you can enter a drawing if you catch a snakehead to win money. For our method of control, should we deem that it is necessary, we will recommend that having recreational anglers fish for snakehead on an incentive-based method. So our research questions for the next 3 years, we have 3 different aspects of our project. We want to know, how do anglers perceive the presence of snakehead in the Potomac River? We want to know, what is the minimum effective population size of snakehead in the Potomac? And we want to know, how can we utilize this information to create a model of their spread? And now Skylar will be talking about our survey.
So to answer our first research question of the perception that anglers have of snakehead in the Potomac River, we will be conducting an intercept survey. Now we'll be choosing locations in Maryland to do our intercept survey from the Marine Recreational Fisheries Statistics Survey list of approved access sites. And as for sites in Maryland, we've been given as for sites in Virginia, we've been given a list from Mr. Newhart of possible sites. We haven't picked any specific places, but we'll do that later this semester. And as for the survey itself, we'll be basing it off a Creel survey from the Maryland Fisheries Resource Office, which you have in front of you and in the appendix of our proposal. And we'll use that as a guide to help us formulate our own survey. And our survey will have 2 main goals, our first of which is to find the perception that anglers have of snakehead in the Potomac River. Now there's 2 possible outcomes. Either they like the snakehead and enjoy catching them, or they don't like the snakehead and think they're a threat to the species they normally catch. Now in the case that they do like the snakeheads, our recommendation to state and local agencies would be to keep a steady population so it doesn't spread and go any farther, but to maintain a steady one so people can fish for it. And if fishermen or anglers don't like the snakehead, then we'll recommend that they reduce the population of snakeheads in the Potomac River. And our second goal is to find out if anglers would be willing to fish specifically for snakehead if they were financially compensated, and if so, how much compensation would be required. Now in any case, if they do like the snakehead or they don't, some form of management will be required to keep them steady or reduce the population. So knowing how many fishermen are willing to fish and how much it would cost would be beneficial to agencies when they weigh what they'd like to do. As for the survey itself, we have not completed one, but will do so by April. This means we'll be able to submit for IB approval, submit for more grants, and test and the survey to see how it flows with students and staff, fix any bugs we may have before the summer of 2014 when we'll run our first field survey. We'll do any fixing we need, any bugs we have before the summer of 2015 when we'll run a follow-up survey, and that should give us enough data. We haven't picked specific dates to run our survey because we all have busy schedules in the summer and we don't quite know what we're doing, but we'll do that later this semester. And now Yasmin will talk about genetics.
Okay, so we hope to collect most of our snakehead samples for genetic analysis through the DNR, and we hope to accompany them on trips for the samples. It's still pending IRB and IACUC approval. Then we'll use genetic characterization to determine size, composition, and other factors about the snakehead population. We'll use microsatellite markers to determine the diversity of the population through specific alleles. Allele frequency is predicted using genotypic frequency through the Hardy-Weinberg principle. We'll sample multiple generations at multiple sources. Using multiple sources will help us to measure the fluctuation of the allele frequency throughout the population. Then we'll determine genetic diversity with the microsatellites using specific alleles. Microsatellites are highly repeated segments of DNA that can serve as genetic markers for information in the DNA. We hope to use 19 microsatellite loci from a study done in New York. This would require that the snakehead in New York are related to or descended from the snakehead here in Maryland. If they're not, then we'll have to find our own loci for the fish here. Then we'll amplify the DNA using a PCR machine and run on a gel. The gel results will help us sample each locus, and then we'll be able to compare all of them. Using statistical analysis, we'll determine diversity of the population and relatedness among generations. Then we'll determine effective population size. Effective population size is the number of breeding individuals in an idealized population. This will help us determine how far, how large, and how fast the snakehead population has spread in Maryland. This is essential for management, we believe, because the snakehead is a recently invaded species about 10 years ago, and therefore it probably has low genetic variability here, and the genetic population size is currently smaller than the actual population size. We'd like to understand the variability, which is essential for either control or maintenance or expansion of the fish, and then we'll know whether or not to make the genetic size bigger or smaller, and we can inform agencies on how to respond accordingly. So throughout the semesters, we'll be forming subcommittees depending on what we're doing that semester, and we'll be rotating those so every member on the team has a chance to experience different parts of the project. We'll have a lab team, which will be in charge of the samples and the PCR and the genetic analysis. We'll have a survey team, which will be in charge of going out and doing the surveys and completing the survey on the anglers. We'll have an analysis team, which will be in charge of using MATLAB to analyze our data after the surveys. And we'll have a grants team, which will be applying to grants and making sure that we're approved for IRB and IACUC. So this semester, we hope to finish our survey by April, and then we'll apply for IRB approval or exemption, and we'd also like to have IACUC approval, and then we can move on with applying for grants. So our end users are the Maryland DNR and the Maryland Fisheries Resources Office. We hope to be able to create a management plan for the snakehead based on whether the snakehead is considered a threat or an opportunity for anglers. And this is our timeline for completion. I'll just go through that now. So we're currently in our second sophomore year. We're defending our proposal right now. Then we'd like to finish it up and be able to make any adjustments as needed depending on comments. We'd like to apply for grants, and then we'll conduct our intercept survey in the summer and hopefully be able to start collecting snakehead tissue samples at the end of the summer. Next year, we'll defend our project at junior colloquia in the fall. We'll start genetic characterization in the fall and statistical analysis. We'll conduct our follow-up survey possibly in the summer if needed, and we might collect more samples if needed as well. Our final year, senior year, we'll be compiling survey data. We'll be drafting and writing our thesis. We'd like to complete characterization and analysis for year 2 if we do that again. We'll defend our thesis in the spring, and then we'd like to present our results at a conference and maybe even get published. And now we'll open up the floor to questions.
Hey, great, very good. Traditionally, we like to give our visiting experts the first salvo of questions. And then we tend to focus on more picayune things. So you do have one yet there, okay? Yes, you have to have that one. Okay, so let's let our experts kind of get going, and we'll make sure we have our mandatory questions get asked.
All right. You want to start? No, we can put bottom me either way.
I just want to say good job with the presentation. I thought it was real nice. It was well put together. It was very different than your proposal, which I struggled a lot with. It was, not typical of a lot of research proposals that I've reviewed or written for grants. It's a little bit long, wordy, and didn't have the nice specific points that you guys mentioned here through the talk. So I found the talk very refreshing, so thank you for that. I guess Josh and I did a little discussion on your work last week as well, and he and I both talked a little bit about it, I think, but I just wanted to really quickly, preemptively mention that DNR is not giving $200 per fish that's caught, right? Okay, so there's no bounty on the animal. Okay, just to clarify, because I didn't get that from the proposal. And to ask a question about that on a survey form, such as, how much will it take for you guys to kill the animal, it's nice. Josh and I both have different opinions on this. It's good information, but there's no way in hell that the state is going to give money per fish caught. Not to mention it's illegal for the Department of Natural Resources to give money per fish caught. It's illegal for us to offer a bounty. It's not illegal for us to have this raffle program, which is what we had. So we've had some incentive programs. I guess my just to get to a couple of questions here that I have for you. site selection. You guys are going to tap the MREP database. Now, I'm not inland side, so I don't. My understanding from MRFs and MREP come from our in-house people who work with striped bass and things like that, creel services. My understanding is MRFs got folded into MREP, so it's now the MREP survey. And when I asked my people about sites on the Potomac River from the MREP, she kind of laughed because it's a marine-based survey, and you probably won't get the sites in the tidal areas, tidal freshwater areas that you'll need from the MREP, right? So did you guys, did you guys look at the MREP access points?
I mean,
am I wrong on that?
The last time I looked at the Murphys website, or I guess MREP now,
Right.
Was last semester, and I just noted that there were some sites on the Potomac River. I didn't really look all extensively quite that honestly.
She mentioned they would probably be toward the mouth where, you catch a lot more estuarine, marine type fish. For our end, for northern snakehead, if you guys want to query individuals on what they're catching, what the perceptions are, you're going to need to be further upstream than that. So my suggestion is to talk to Josh, because I think one of the important elements to your creel survey is going to be comparative, because their office did do this survey a few years ago, and since then DNR and Fish and Wildlife have implemented a number of programs that we can debate whether are successful or not. I got the impression from your proposal you didn't feel like they were that successful. I did follow up with people like Chad Wells, and you probably shouldn't cite Josh Davidsburg because he's no longer with us. He works in Office of Communications, and basically that press release is one that I wrote that he funneled through his office, right? Because I don't get to write I don't get to publish press releases. So I don't know if you guys think that they're successful or not. We have put a lot of information out there, and we've had a lot of anglers change their minds on the snakehead. They've also learned a lot. For example, they don't crawl across the land. They don't come out of the water. You guys need to update that life history work a little bit. But I think with the comparative work, there's some power there because you've got a baseline of understanding prior to our incentive programs. Then you can run this creel survey and determine if any of those you can measure basically the effectiveness. I like to think they're effective because I know what the objectives were for those incentive programs, and I like to think, oh yeah, these are working out because I talk to anglers. But this is one way to measure it. So my encouragement is to follow, and when you're, so here's a question for you. If you're going to generate a comparative study, what's the best way to generate the comparative study? How do you set up the methodology for that?
What do you mean for the?
Well, if you know that this creel survey was conducted, when was it conducted?
2008 and 9.
2008 and 9, and you want to compare metrics, say between now and 2008 and 9, what's the best methodology to use to generate a less biased comparison?
I don't know about less bias, but I would assume we would want to keep our survey very similar, especially if we want to compare results. We don't want too much extraneous information that they didn't ask previously. But I don't quite know what you mean.
That's it. That's what I mean. And so how would you choose your sites?
We would have to use pretty much the same ones that they had if possible.
Okay. All right, good. And you mentioned, Josh, go ahead. Talk a little bit.
Just a process before we over to Josh. You guys feel free to take notes so you don't have to try to remember all these comments. I mean, someone can pick up a paper and a pen and jot stuff down. I don't expect you guys have great memories, but.
Is that still filming or no?
Yes.
Yes, okay.
Not off the film yet. So.
No, if we have it on record.
Yeah, you will have it.
Okay. Digital record. Yeah, good.
I will say, nice job on the presentation as well. Like Joey mentioned, we talked a bit, so we had a lot of the same ideas. And yeah, you're right. For the creel survey, if you want to use this as a good start, that would be great. I would suggest that you narrow your focus to what you really want to ask. Some of these you know might not pertain to you, and also if you're going to ask about an incentive-based program, think about how you're going to phrase that question. It's really important to have this list, obviously, of questions, and you want to vet it fairly well to make sure you're asking the right stuff and know what analyses you're going to do in advance. I know in the proposal you mentioned certain things you want to do, but I'd like to see that a little bit more narrowed down. And I know you mentioned you'd have it by April or something. So that's good. I wouldn't worry about, you mentioned about influencing anglers with snakehead. You want to ask them what it is. If you want to ask them what it is and point to a picture, that's actually really good, actually, because you know we would just ask them if they knew what a snakehead was, and just because they say yes doesn't mean they know what it looks like, which is important. But I don't think you're going to bias your study. As soon as you ask them the first question about snakeheads, they're going to know, because it just gets people.
We're thinking about showing different pictures of different fish and asking them, do you know what this fish is? Do you know what this fish is? And then include the snakehead. So I don't know.
Okay, I mean, I wouldn't worry about bias in it.
Yeah.
You're right, the bias is going to come from all the media that's been received, all the media attention that was received in the past. So
Yeah.
that's the kind of stuff that's going to bias your study. And I mean, that you have to address after you ask the questions and think about the results. So I wouldn't worry about that. Like I said, I can talk to you about how you want to structure the survey and where you want to do it. If you want to do exactly the same boat ramps, I mean, that's fine. That would benefit us as an agency. We'd be getting comparative results. Joey's right. You don't want to follow downstream sites on the Potomac. You're just highly unlikely to come across anglers who are catching snakehead out of Point Lookout or something like that. You want to think. I'd like to see the methods more fleshed out. You got to think about how you're going to weight these interviews. You're more likely to encounter anglers at one point in the day versus another. You're going to be more likely. Some ramps are going to be busier than others. So I can tell you when we did our survey, we weighted. It was a random, stratified random design. We weighted interviews by time of day, AM, PM, and then also by ramp. And we chose the most popular ramp that got heavier weighting, so we're more likely to interview there. And so you need to think about doing that kind of stuff. And I can go over with you what we did down the road, and just to let you know how you might want to structure that. And the good thing is you have the time now, if you think about it, to really, you can, like I said, fine-tune these questions. Some of these are going to be really relevant to us as a federal agency, but also to the states. Like, do you ever plan to fish for northern snakeheads? That kind of stuff. We can see if attitudes have changed with trying to increase the amount of press that has been out there about snakeheads and seeing how they feel about it. That said, what do you guys think? You said you're going to use this as a structure. Have you thought about some of the additional questions you might want to ask? I mean, you talked about incentives and stuff like that. Do you have some ideas how you might ask those questions today?
We haven't quite got there.
Okay.
But
We're planning on it in the next month to start developing those questions and thinking about that.
Okay. Well, Josh and I talked a little bit about this, and I talked about it with my boss, the assistant director. And I think on our end, we were not comfortable at all about asking anglers how much they would need in order to kill snakehead, because for us, we dealt with a lot of miscommunication. I personally dealt with a lot of miscommunication when that stupid $200 bounty thing got blogged about, and then Fox News and every media corporation from across this country picked that up. I was dealing with people out of California. People wanted to come out here and do movies on this because we were doing this $200 bounty per fish. So we're very sensitive. I'm very sensitive. That was a lot of wasted time that I spent dealing with it, and I just didn't want to go down that road again. But Josh had a decent idea, because he said that there's some precedent for federal agencies to pay contractors to go in and harvest invasive species like nutria.
Yeah, and you can think about, like I said, comes into the phrase, and you can think about, you don't want to, if you want to ask them, will you accept $50 or something, that's a little bit more direct, and they're going to potentially expect that, or they're going to at least think in their heads like, oh wow, they're thinking about paying us this much.
And we're not thinking of paying them that much.
Yeah.
There's no way.
So there's ways around that you can say, would you be willing, to be hired to fish for snakehead or something like that. You can also, or, you know, we mentioned that, I thought about, you could also just make sure to be clear to them that, you know, you're not getting paid just yet. Because obviously that's going to be a lot of money. Just so you know too, Bass Pro did this back in 2003 or 4. They actually were paying anglers, I forget, 20 or 50 bucks until a guy showed up at their door with, you know, hundreds of snakehead and said, you know, where's my money? And that put an end to that program. So this has been done. And so think about those kind of problems too when you're thinking about, you know, moving forward with this. Now, social control that's a great idea. We've talked about this a lot. You know, us as agencies, we can't get out there enough. And there's a significant amount of angler effort out there. So that's really important, and we want to stress that. And this kind of survey will help further that along. But with the money, like I said, the state's real concerned, so you want to be careful. And people are fishing from Virginia as well, and they might expect that from Virginia, so you want to really be careful. Think about that. Like, it might just be as simple as saying, well, do you want to be hired as a contractor to fish for snakeheads, or if they would be allowed to sell them commercially. Now, I might have missed it, but you said they weren't allowed to be sold commercially. In Maryland, their commercial fishermen can sell snakehead.
Yeah.
Recreational anglers can't. Now, that would be really interesting to the state if recreational anglers wanted to sell their snakehead catch. That would be a good question to ask.
I already know the answer on that one.
Well, it's probably going to be yes, but
Yeah.
that is something that the state could actually take some action on, and that would really make a worthwhile decision on.
That's a good
if
point.
they allowed recreational anglers to sell their catch, which would open up a new market.
So could we be asking them, would you like to be selling your snakehead catch?
You could ask them, something like that. If you could, would you sell your snakehead catch, for a profit?
So many of the tagging programs use an incentive. They don't use a bounty system. And I think they were thinking more of an incentive program where they say, you give us a snakehead, we'll put your name in the drawing for a hat.
Right.
And one of those drawings will give you a $1,000 prize. Is that conceivable?
Yeah, I mean, the state could get behind those raffle type projects. When we dealt with this with our Attorney General, back when we were deciding whether we could even do a raffle, our legal folks said that it's a little bit of a gray area with that, but they allowed us to do it. So I think we could, that information could be meaningful to the state agency because the state might be able to do something like that. There's just no way that legally we couldn't even give $1 a fish. Remember the going rate, what is it by pound? It's like our commercial guys are getting like
$3 or $4 a pound.
Yeah, $3 or $4 a pound. So it honestly makes no sense to even give $50, right? Because a typical snakehead's what, 8 pounds? So you're talking maybe $24, $25 per fish. Why in the world would the state give more money to harvest a fish than what commercial guys getting to sell? So, but they do tag them, and they get free hats if they report them.
Yeah, I mean, and that's incentive based, and from the federal side, I know we used to give $100 per sturgeon that was called in, so.
But that's not
there's
a
some
bounty.
precedent there.
You're not killing that, and that's the point. They're reporting the tag, and that incentive is fine, but if we can't fund someone killing a species, we just can't do it. It's just
Do you
illegal.
think someone like Bass Pro Shops would be more willing to do a raffle-based system? Because I can see they get burned by, people who just say, well, as you said, $50 a fish a weekend, I can make a lot of money
Right.
on this.
Well, that's where the $200 gift card came from, Bass Pro. And then this past year, we're actually changing it now, but this past year we merged our snakehead contest into a volunteer angler survey, and that was incentivized with a $200 Bass Pro card. And now, moving into this year, our director wants to redo this angler log contest in addition to our, here's your scoop, in addition to our offering a state record for snakehead this year, as well as
Just tweet that.
It's actually a state record. We have it's a state record for an invasive species. So we're still molding that concept of killing the animal because they're invasive. So we're doing that, and then folding it into our Maryland Fishing Challenge as well. So there's going to be some other incentives for killing the animals. So we can do legitimately those types of things.
So let me ask you guys a more philosophical question. This is the nuts and bolts. Let's say you do this angler survey and they say, yeah, we'd love to participate in a raffle-based system, and we think, you know, $1,000 would be great, and we'd be willing to go out and catch snakehead. And what happens if they turn around and say, well, no, actually, we quite like catching snakehead. We want to encourage the state to manage this fishery because it's a good thing. Is there a philosophical point where the state agencies have to say, we really don't care that you like catching it, we've got to eradicate this fish because it is an invasive species, it's a bad thing? Does the angler attitude really matter here?
I think it does, but also I don't work for a state agency, so I can't really, I don't know policies like that, so I wouldn't know how much information like this, if successful, could impact policy. We're just
So
hoping
put on your hat as a scientist.
Mhm.
Is an invasive species such a risk that regardless of social attitudes, every effort should be invested to eradicate it? Is there ever such a thing as a good invasive species?
Say no, huh?
No.
Well, isn't the largemouth bass, or is it the striped bass, that's the naturalized species? That was an invasive species that's now,
It's largemouth.
Yeah.
that's largemouth.
Stripers are not native.
Okay. So the largemouth bass is now a naturalized species that just, it's helpful to the Chesapeake Bay economy and the area around it, right? So if
So
that's,
it might be helpful to the economy, but is it a good thing? Is the state agency, is science pressured to keep these ecosystems natural? Should that be their goal?
I feel that they're pressured to keep these ecosystems stable and healthy. And in the event that like an invasive species is a real threat, a real danger, like I believe the northern snakehead is, then I believe they should have some sort of management protocol to at least keep it managed, if not eradicate, because I think it would be harmful to the ecosystem and just set off chain reactions.
So the Chesapeake Bay Foundation will often say, we want to restore the bay to the time when John Smith arrived.
It's true.
Is that a legitimate scientific goal?
well,
It's not.
the environment has changed so much since John Smith arrived. So they have to be able to, like, I understand where their idea is coming from, but at the same time, they have to be able to understand and legitimize everything that's going around the Chesapeake Bay in order to maintain that stable environment and to maintain the Chesapeake Bay in the environment that it's in. Rather than, because you don't know if they try to restore it back to what it was healthy then, but we don't know how it would be now with the air pollution, the water pollution, the real estate around it.
And to kind of follow up on that line of questioning, your team as an independent, academic, aloof, ivory tower, non-beholding group of researchers, you spent a lot of time in your proposal talking about how you could provide support, provide backup, provide information to agencies, whether DNR or Fish and Wildlife, to make management decisions. But the next question was, is that your goal? Or is your goal to discover new information and lay it out there to see whether the agencies can utilize it? Or is your goal to get information to support their mission?
In my mind at least, our goal is really just to find more information. We were hoping that, maybe this information can be something that these agencies can look at, and maybe it can be of use to them, and maybe it won't, but having an impact would be great, but really just finding out more about this fish and how it's affected this area is really our goal.
When you revise your proposal into your thesis work, that tone of supporting the agency mission is pervasive in your writing. You want to make sure that it's mentioned as a possibility, but not the ultimate goal of your work.
Yeah, I think we did more on our presentation here than in the proposal because we thought about it after. And not being objective and
Works
things like
evolve,
that.
right?
So that's why we say we'll have to change it after.
You want to get some questions in?
Yes, so how did your team explore, assess, and integrate scholarly resources into your proposal writing process?
Google search.
What's a snakehead?
We pretty expansively utilized Maryland's, especially the library resources, to help narrow our focus and find research that was applicable. We also reached out to a graduate team, or what was once a graduate team in Virginia Tech, where they did some research on snakehead, and they did tagging, I believe. And we asked to see some of their lit review, and they gave us access to a couple translated Chinese sources that they found helpful. Their focus was a little different than ours, but it was still good to have that kind of information. And that's where we got most of our information. Of course, I didn't find every single article, of course. My whole team, we all worked together. But that's where I believe most of our information came from and where our knowledge base came from.
Josh, you have a fishery management plan,
Yeah.
correct?
Yeah, I was gonna get to it, but.
Talking about referencing.
Yeah, I brought you a copy of the latest, there is actually, hopefully will be finally, National Control and Management Plan for snakehead. It's actually for 3 different species, but the northern snakehead is really pervasive in here. So I brought a copy of it. If you guys, I can send you an electronic copy if you want. Ideally, that'll actually be published. It has to go to Congress, but ideally later this year it'll actually be on the Federal Register and be an official document. But this could really help. You look at that, there's actually a lot of where research needs to get done. There's a lot of what has get done. This would be good to use. There might have been some versions out there on the web. I can't remember if you cited them or not, but this is the latest update, and hopefully it'll be the final one.
It's an extensive literature review.
Yeah, so
Extensive.
that should meet a lot of those needs, because I have some electronic. I also have printed out comments I made on your document and the general paper that are a couple paragraphs I wrote about it. I'll give it to you guys too. I can send it to you electronically if you want. Just let me know. But that said, the lit review does need some work. Especially you cited a Mason 2003 article. That was largely an opinion article. I know it was published in like a Nature Letters or something, but it was an opinion piece. So with a lot of hype. There was a lot of hype when it first came out.
A lot of hype.
Walking across land, eating everything. Watch site and that kind of stuff. There's been a lot more done since then. So that, just a general comment on that. And like I said, this will really help your lit review. This
You guys
management plan.
feel like your writing style, when you're taking a technical writing class, do you take that here at College
Technical
of
writing
Law?
is a junior class for the most
Yeah.
part.
So it's coming?
Yes.
So it's
Already
coming.
Coming.
Okay.
So
So
hopefully.
you guys get that in science-based writing, you've read papers, I'm sure, peer-reviewed papers, primary literature, gray literature perhaps. You realize that when folks write these, they tend to not be sensationalistic. Now, I appreciate the fact that this is a proposal, in which case you need to drive your passion to the funding agencies so they understand not only your competence, but that you're passionate about the work. But I got the impression you guys were really trying to sell me that snakehead or demon spawn.
So we wrote it, and then Tom asked us, okay, what if anglers think they're a good thing? Then what do you do? And then we were like, oops. I think one of the biggest changes we need to make to our proposal is changing like the tone of it and making it way more objective and like a proposal. And I think that's definitely one of the things we need to work on, that we realized after it was submitted that we should really work on that. So
Let me follow up with what Tom is talking about. So if an angler does, some of you will be conducting this creel survey. And I've known in the past from creel surveys, creel clerks can be somewhat timid or occasionally lazy, and I'm sure that doesn't apply to any of you.
I can
But
show
we-
you it doesn't.
That's very good.
which part?
But you may become intimidated when you get out there. These are anglers. I'm sure you guys have dealt with them. You might be anglers yourself. You guys fish? Good. You got 2 anglers out there. Maybe you guys should be doing the creel surveys then. These anglers can be a little salty sometimes. They'll look at you. And especially if per chance it comes out that you're working with DNR Fish and Wildlife, you'll get an earful. as to why we're screwing up the resources. So it's very possible you'll ask them a question and it'll be, well, do you think snakehead are good for the environment? And they'll be like, yes, DNR and Fish and Wildlife are screwing up the environment. Do you guys, you guys don't intend on arguing with them, do you, or engaging? Your goals are going to be strictly recording the information that they give you, and then you can analyze it down the road and say whatever you want to say about them,
And I
right?
think also staying far away from DNR and not mentioning it at all would be good.
Yeah, no, I wouldn't say that. There are, depending on where you are, like if you're operating at a Smallwood, we're out there a lot. And they see us, they engage with us a lot, and we talk to a lot of them. I would say we have good rapport with about 90% of the anglers that I've dealt with. There have been a couple of jerks. One who apparently threatened to bring out his gun. But, so I do take some things seriously. I don't joke about it. I've had biologists in the Upper Bay last year pulling out bass for our hatchery program, and they were threatened with a gun. I was threatened with a gun while I was down at Friendship Landing. So I think that there are elements of that, but generally speaking, 90% of the anglers out there like DNR, and they like Fish and Wildlife. They think we're doing a good job. So you can mention it, perhaps, if it comes up, but just don't argue with them. Like if they, I guess you got the tone of your proposal was to convince folks to hate on snakehead. I just don't want you to be on the ramps asking the questions, convincing them to hate on snakehead. That's not the place for it. That's our job. It's our job to do it. You're feeding us this information. I mean, it's possible some of this is going to be academic, just collecting information for the sake of information collecting. But be aware that when you come out of this think tank, even Tom Miller gets funded, like a lot of money from state and federal agencies. We all get funded, and we have to answer to those entities. So there are certain management questions that he has to answer. When I was working at the university, there were certain management questions I had to answer. So it can't all be think tank. It can't all be esoteric type stuff. I'm going to publish in Science and Nature and become very famous. You can't just always do that. Some of it.
Once in a while is fine.
Once in a while is fine, but you do have funding. You do have to answer to NOAA, Fish and Wildlife, DNR, and so on and so forth. Is it too early to talk about the genetic work?
No. I want to make sure everyone get a chance. The librarians have a bunch in there.
No, I mean, I didn't know. The only thing from a library side, from citation side of things, just sort of a nitpicky kind of thing with your citations, the format of it, just double check that when you cite them in the resources list, they should be single spaced in that, like each individual citation.
I think actually APA 6 calls for it to be double.
No, it does. As far as I know, it's still single.
No.
Were the citations right this time?
They're better.
And just following up with where you get those citations, if you're having trouble finding, the scholarly resources, using the reports and looking at the references can be a good source for original research, and coming to us, that we can help direct you to databases that you can look in.
What does ND and NP mean? I wrote these are not standard because I have no idea. I've never
No
seen
No
it.
data, no page.
Oh, okay. Usually we see unpublished data, unpublished, or
Right.
just personal observation. But
Yeah.
Indian.
How many is the question? Or should we continue?
No.
Let me say one, I don't know if we're gonna go off Chris, let me say one thing. Like I mentioned about narrowing down the focus. You're only gonna have a very limited window in which to talk to these people. You're gonna get a few people who are gonna wanna talk to you for an hour. They're gonna be annoying. But you're gonna get very few that are getting off their boat, they don't wanna be talked to. So think about that when you're making these questions. Like I said, if you wanna use this as a template, that's good. Get the important ones you want. Perhaps, I was just looking like what % of the time do you fish from that ramp? It might not be that important to your project, so. You're gonna want yes, no. You're not gonna wanna give them many choices, like, don't give them an open answer question. Like if you're gonna, however you phrase your incentive-based one, if it's gonna be a contractor, don't say, well, how much an hour would you wanna get paid? You wanna have defined options, which would really help in your analysis too, so. Just think about that when you're putting these together. And like I said, I can help you work on that too. So.
Okay. If we want to shift first to the genetic part of it, I'll start off with a question about, you have a lot of your baseline information comes from the study from New York. And the big assumption is that genetic pattern will be a good enough benchmark to compare the Potomac genetic pattern to. Is that a big leap, a small leap? Are you confident in that leap? To me, that's a big leap. But how confident are you in that?
You mean as in having the same common source, or?
Right, if my understanding is correct, and correct me if I've misinterpreted, you're trying to use the New York genetic sequence as the base you can compare the Potomac genetic sequence to.
So that's the one that we found.
You can go ahead.
Oh, you know more.
So it's for the microsatellite analysis, it's the 19 loci that were found in New York, and we're going to we want to use those 19 loci because we do believe that they will be similar enough to the ones here, and we want to use those 19 loci in order to analyze these fish because those are already found. If we find that it's not working, then we will have to go back and find our own loci in the snakehead that are in the Potomac River.
Okay.
So it's kind of an ideal situation. We
Yeah.
just said this information's out there. It's totally worth trying. I think it's relatively feasible that will happen. But if it doesn't, it's a road we're prepared to deal with.
Okay, but think back to the physical pathway which the species was introduced both in New York waters and in Maryland waters. Where did the fish come from, and what's the possibility they had the same genetic base from their origin? How did they get into Crofton Pond, and how did they get into whatever that pond in New York was? Where did they come from?
I read the.
Well, Crofton, the ones in Crofton there's a lot of debate just because it was emphasized earlier, it was a very sensationalized story, so there's a lot of different information, but the general theme seems to be that somebody had these fish as a pet or in an aquarium or something akin to that, and decided that he didn't want them anymore, or she, whatever, and so they dumped them in Crofton Pond where they were able to then move forward into the Potomac River system. So we don't know necessarily like exact specifics, but from what I understand in New York it was relatively the same, where somebody had these fish and didn't want them anymore, so they just figured it would be fine to just like dump them in a waterway, which has obviously resulted in...
Okay, well, you're on the right track. So where does aquarium stock come from?
I'm sorry?
Where does aquarium stock come from?
Asia. And so we hope that back in Asia, they're closely enough linked to when they came over here. There's
Right.
something in common.
So the step in between, are there 3,000 importers, or is it one importer?
Yeah, so that's what we're trying
Yeah.
to figure out with we know of this Crofton, Maryland introduction. We don't know if like other people in Maryland were like, oh, there's snakehead proliferation, I might as well just dump my fish, my snakeheads in the river too. So we're also that's why we're doing the microsatellite analysis and trying to figure out the density of the population and the genetic characterization to see how many origins there are, how closely linked they are, because we believe that if there is only this one origin, there shouldn't be that much genetic variance, especially from even between Maryland and New York, because as Skylar said, if they're closely linked enough, then even in New York, the genetic variance should not be that much. And then compared to Maryland, those 19 loci should be close enough. So we're trying to figure out, were there different origins, what is the genetic variance, because we don't know that.
Yeah, I totally agree with how you're thinking this through. But what do you think about the practical side of it as well as the genetic side? For example, do you need a permit to import snakehead as an aquarium species into this country? I don't know. Do you? I don't know if you do or not.
A permit to, well...
Who can import them?
Yeah, nobody right now.
Right,
Yeah.
you can,
You
yeah,
can get a permit
it's still
It's
illegal to
banned, yeah.
possess them.
unless you have a permit to possess it.
Right.
Okay, so the avenues of entry may be narrow, or they may be
Well-
many, I don't know.
You're not gonna get that permit in Maryland.
Yeah, I don't know how much should lead you on or just answer your question for you.
I
A
think-
lot of this work's been done already. check oral and weight. I know you didn't cite that in your proposal. They worked at Smithsonian. They did a large amount of genetic work in 2005, and they published it. That citation's probably in that document. You cited one of the King and Johnson papers from the New York. He also published stuff on the Potomac population and talks about introduction and kind of population structure. So when you go back and do your lit review, you'll see a lot of that stuff, and that'll help refocus your genetic work on what you want to get done. That way you're not redoing work that's already been done, and you can make it a little bit more relevant. I can tell you, the New York population and Potomac, they're two different introductions. They're not related. But King's already done the work with the microsatellite DNA, so you can still utilize his work in that end.
And just by way of clarification, Crofton pond introduction likely occurred somewhere around 2000. They were found in 2002. The Potomac River, they were first found in 2004, and that was a separate introduction. The Patuxent River co-joins with Crofton ponds right through high floods or whatever, but that's very well isolated from the Potomac. So two different groups of animals there, likely multiple. And based on Tim King's work for the Potomac River, there were multiple introductions into the Anacostia, probably through Kenilworth Gardens, right? You guys should be familiar with that area because it's right around here. And that's likely the source population for the establishment of other pops within the tribs of the Potomac. So just keep that in mind within your framework. They're just very different. And plus, like you said, the microsat work for the Potomac has been done, so those markers exist. They should be successful, right? So you're going to run these out on gels?
Yeah.
Is that correct? Okay. So you're not sequencing?
No.
Okay. And you're going to use an acrylamide gel or an agarose?
Agarose.
Do you think that that's going to, well, from what I understand of genetics, and I did a little bit while I was at Mississippi State, didn't publish, I'm not an authority, right? Acrylamide tend to be the gel of choice for this kind of resolution that you're in. We're talking about a few base pairs, and I get that the microsatellite region is, a hot spot. I get that. But do you think the agarose is going to give you the information that you need for genetic diversity?
Well, I worked in a lab, so I'm not entirely sure, but I thought acrylamide was used for proteins, and then agarose is more for DNA, because
Yeah, I used acrylamide for running DNA. Actually, I was working with the mitochondrial DNA, which is what I ran out on acrylamide, but that seemed to work too. There's a paper that I just got online about it, because I was kind of curious about whether you could even use acrylamide to really, I'll just tell you, most of the microsatellite work that I'm familiar with, now I haven't done that, I haven't used microsatellites as a marker before. Most of the work that I've read about while I was working on my PhD, I worked with geneticists, we worked with Tim King. What Tim King's done, it's all sequencing. They don't use gels. So Tim King's used sequencing to get his estimates of genetic diversity and such. I get that it's very expensive though, right? I don't even know if you've got a sequencer here on campus, let alone a team to operate it, because learning it is beyond the realm of what you can do within this time frame you're given. So I get you're going to be running gels. I just want to know whether you think you'll be successful or not. I mean, I know you hope to be successful, but have you read enough to know on using that technique for microsatellites? Have you read enough to know whether you're going to be successful using that or not? Or have you thought about any preliminary work that you can do to even determine if you can isolate the marker with, some primer sequence and then run it out on the gel? Agarose is great for detection, right? At the very least, can you do that, and then maybe perhaps I don't know, maybe you could use a DNA ladder on an agar-based gel, that'll tell you something about
Yeah.
genetic diversity, right? Are you planning on any of that kind of preliminary work prior to the proposal work?
As we move more towards that aspect of the project, we're definitely going to be doing more background research so we understand more about the process, so that we can turn what's normally kind of,
from what I understand, a cookbook procedure, pretty much do A, B, C, into something that hopefully will be more often successful than not. Hopefully, yeah. So,
Okay.
yes, that's something we're going to be looking
using
into.
test primers though, just to make sure that everything's working.
Okay, well, maybe you could talk to Tim King, he might be able to provide you some primers. I mean, they're really cheap, right? I think
Yeah.
they're
Yeah.
$10,
They're-
but
They're like $7 for like a 20.
like enough,
Yeah.
right, yeah. But maybe, you could talk with Tim or something if you, do you have a geneticist you're working with?
No.
Not yet, but we can get into contact with them.
Yeah, I think that would be a great idea. I mean, it's very, I get conceptually, genetics makes a lot of sense, but conceptually, so do creel surveys, and both are very difficult to implement. Genetics work takes a lot of patience to get done right. And you guys are talking about publishing, I just want you to get it done right, because if you go to a journal with, haphazard stuff, then you're gonna get disappointed, so.
We definitely need to read the King studies that you guys talked about.
Yeah, or orals, yeah.
Yeah,
Right.
and then we're right now trying to find a lab that we could possibly be using next year.
Okay.
And then I think we should probably try to find also a geneticist on campus that can help us out with that.
So you do in fact have two geneticists in your course, you just haven't met them yet.
Yeah.
So you'll be fine with that.
Okay, very cool, very cool.
So Lewis Plow at Horn Point has volunteered to help, and Matt Hamilton at Georgetown has also volunteered to help.
Very nice, very awesome, awesome, use them, learn as much as you can.
I'll
Josh?
ask a more, I'll ask a more philosophical question, I'll make everybody else happy. How do you think, when these genetic samples, what is your time frame for collecting these genetic samples in terms of seasonality? And how do you think that's might affect your results? Think about spawning time for the snakehead and when you're getting these samples, and how that might influence your results.
Didn't you guys talk a little bit about migration?
Yeah.
Right?
Yeah, so we were really hoping to get started in the summer, and that's when we wanted to start doing a lot of the collection. We haven't looked extensively into it. We haven't picked exact times or anything like that, as Skylar mentioned during our presentation. But as we start working on IRB and IACUC approval, that's something that we're really going to be looking into.
Okay.
Well, what would be the difference? What if you get fish from May before the snakehead have spawned, and you get some you're gonna get some samples in September after they've all spawned? I mean, will that change your results, and how might it change your results or influence the genetic diversity that you're seeing?
So we were planning on being consistent with the time that we collect the samples, so we said around late summer for this summer and late summer for next summer if we do that. But we haven't really thought about if we should try to think about a different time. I think we were saying like August time for collecting samples, but yeah, I think we definitely need to think about what would happen if we started like, if we thought about another time or.
Let me try and lead you further down the path that Josh is trying to open the gate for. You mentioned something about generations. What will have happened between summer and the autumn time?
New generations.
Yeah.
So what happens with each generation? What could happen with each generation?
More genetics.
Just
More diversity.
tweaks
More
to, yeah.
diversity.
Yes.
Or
Less.
Or, yeah.
it could be a change in the change.
So you're saying we should run a comparison between, say, early summer and end of summer?
No. What Josh is saying is that when you sample can matter, and your comment that it needs to be consistent is the important point.
And to put the undergraduate spin on it, when you're sampling a biological system, and you know that temporal timing of your sample is important, it doesn't necessarily conform to the academic calendar or to the summer break calendar. Or you have to make a determination of when, given all those factors into consideration, when can you get it done and when's the optimal time to do it, and justify why.
And how would you justify why? I mean, Josh and Tom kind of talked a little bit about how do you justify why you're going to sample when you're going to sample.
We have talked a bit about this, so we know that we want to do, well actually I believe you mentioned it earlier, Tom has talked about random stratified sampling with us, and he said that that's really important with the survey, but it's also important to keep consistency with when and how we collect the samples of the tissue, and so that's, if that answers your question.
It's an answer, it's not the answer I was looking for though. I mean, the answer with respect to justification for the genetic work, it's not about you, right? It's not about what's best for you. It's about what's best for the study. And how do you know what's best for your results? How do you know what's best for your study?
We look at other studies.
It's one thing, but you do have to understand the life history of the animal, which is kind of what Josh was hinting at too, and what Tom was talking about with generations. This animal moves around within its own, within a year, right? And that movement's going to bias your genetic work, and you want to try and remove that aspect of bias because you're interested in identifying genetic patterns as well as you can, right? Do you know anything about the life history of the animal?
So for the spawning, correctly, do they come out to deeper water?
No, they usually
Or they come
go
back in?
upstream to
So,
smaller waters.
when we were first figuring out, we didn't know who was going to get our samples. We didn't know if we were going to have to get them. So we chose, we were trying to choose when they spawn, so they'd be around us. We wouldn't have to go to deeper water to find the snakehead. We have a likelier chance of getting it in shallow water. But we don't know how that would change now that we could possibly go out with other people and collect
get it.
samples.
Okay.
That was our reasoning for picking the summer. And plus, it just coincided with us being off school.
Well, intuitively, I think that's the best season actually for the animal to get that.
I think your late summer is actually probably a good time to do it. It works for you guys because you're off, but you're also, that's the time where they're moving the least. So you're going to try to avoid that bias. That said, you know, you're getting them, if you're getting some of the samples from us, if you're getting all of them from the agencies, you know, then you might be limited in your sample size in that. So if you want a bigger sample size, you may have to take them from a longer time frame. And, you know, that said, if we're killing fish, obviously we can hold them and freeze them or whatever for you guys, or you guys come pick them up. But, so, you know, you might have to take them from a little bit broader time scale, but then I just want you to think about that when it comes time for the results and how you're going to analyze that. And that said, this is a good segue into, well, where are they going to come from? I mean, the Potomac is a huge place. Where specifically are they going to come from? Do they need to come from a wide area? Is one tributary enough?
Well, I think we were talking about maybe asking anglers, because I think there was some, there's issues with IRB about asking people for their catches or for samples of them. So we were, I think, I don't know, was it you that suggested, Tom, about maybe having samples of that, but then it goes into how different will they be from different sites. And I don't know if we'll need to specify different sites that we're going to be collecting, because I guess it is.
We would definitely want to get as many sites as possible.
Yeah.
To diversify.
But it is.
That's right. That's like every biologist's dream.
Yeah.
However, living in the real world,
Yeah.
what your objectives are to estimate effective population size and genetic diversity for the entire Potomac River population. That's right. I mean, that's what I read up there, right?
We can always narrow that and try and take a section of the Potomac where we find enough snakehead samples.
I guess that would also depend on where our sites end up being for the surveys, because I guess we would want that to correspond, because we don't want to be asking about snakehead in one place and then sampling from a completely different place, because that just might get us different results and it's not consistent. But I think definitely narrowing it down and specifying it is.
So you learned one thing today that I certainly didn't know that could make your lives a lot easier.
Yeah.
You focused perhaps on having the agencies sample your fish. Okay.
I just need this.
Do you need some water or something?
I have water in my jacket.
Go ahead.
We've focused on having the agencies collect for you. You've thought about having the recreational fishermen, if they had snakehead carcasses in their cool box doing it. What's the other group that we found about today who can legally hold snakehead?
Commercial fishermen.
So you can buy it.
Oh, I would even hate for you to do that.
Why?
We could just get them from the commercial guys.
Well, you
they're on
don't
the,
have to buy it.
yeah,
They're gonna, some of them they're filleting, so you could just get the, you're just doing
Fin
call
clips,
fin
right?
clips, so.
Right.
yeah.
Well, it would be
We
nice
could-
to have outlets to go with.
But, they're just toss, if they just toss that, I mean, of course, if they're selling the whole fish, you might.
You could get them off the bow fishing tournament too. or the
But
fishing
I
tournament.
will say there's only one company, I think now there's 2 distributors selling them, so
Yeah,
it's not hard to find them. And the guy, we know the guy that his company sells them. And
Yeah.
I'm sure he'd be more than willing to write it off for charitable donation or something, so we can give you his contact information if that's the route you wanna go. That said, you're not gonna know where the fish are coming from most likely. That would be the downfall there.
So
You might
people-
lose that spatial.
Yeah.
But you would if you went to the commercial guys.
No, you don't know what necessarily the
Yeah,
harvesters
they
are
don't,
getting.
yeah, not necessarily.
I mean, the harvesters could be getting them out of Mattawoman, and they could be getting them out, and they mix them all up.
Yeah, it's gotta come from Maryland, though, that's for sure.
Yeah.
It will come from Maryland, because they're not allowed to sell them in Virginia, so.
Right. They all come
I do.
Right.
Yeah.
Right.
So, actually, I thought about that on Friday, thinking about this. The recreational anglers, if, this goes back to, I have a lot of comments about just narrowing your focus, really hammer down your objectives and what you wanna do. If you want the effective population size of the whole Potomac, that's a really lofty goal, and you're gonna need them from a wide spatial distribution. And the recreational anglers could be your target for that. Because, they're not all fishing in the ramp that you're gonna interview them at. They're gonna have to know where that fish came from, ideally, if you wanna get at things like population structure. So it is possible. Now, if you encounter hurdles, you may, apparently, I don't think that'd be a problem, but you're very likely to come across anglers at times that have snakehead that, if you say you wanna cut their fin, they're probably not gonna care.
But the other, thing that you might wanna consider, just FYI, Josh and I, our agencies are doing work on Pamunkey River, which is on part of the Potomac, this year to estimate population size and assess the influence of fishing pressure. So our goals are gonna be to go out early this year, estimate population size, and then through the summer, kill a bunch of fish using electrofishing, killing as many as we possibly can. And then at the end of in the fall, we'll be estimating population size again. We're using tags, right, for tag recapture work. Y'all are more than welcome to have any of those dead fish that we pull out.
You should be smiling big now.
If anything, I think it would be fairly useful for us, because not only can we then estimate what the change in population size is due to fishing pressure, we can also estimate what the change in effective population size is due to fishing pressure. And because we know what fishing pressure is, because we're exerting it, I think it becomes a useful component of the study. So if y'all want,
Yes.
Please. We will take your dead fish.
we will be more than happy to keep them for you. So you can either have the whole fish if you want to do the otolith work. Man, it's an extra part of the study, and Odenkirk is a success with aging with otoliths, with snakehead. Virginia has not had as much success. I think it was Virginia Tech. Wow,
I think we've tried contacting him.
Yeah,
Yeah, we
Odenkirk
have.
is not very
He's not very receptive.
open or cooperative.
Who, Odenkirk?
Yeah.
He's a busy man.
Yeah.
But yeah, his otolith work, he's published it a little bit. So we just talked to him last month.
Yeah, he's not sold on it.
I'm not sold on
Yeah,
it.
not me.
But we've got to
More
figure out
samples,
to
yeah.
age him at one point. But if y'all want to go down that road, I'm just telling you that the methodology has not been fully developed, and it has not been, those otolith ages haven't been validated. It's not like he grew the fish up 2 years and then took out the otolith to ensure there were 2 rings. He didn't do any of that, so. Yeah, but he did publish it without the validation work.
Has anyone done marginal increment analysis on it?
Not to my knowledge. We're not. Virginia's not. So, yeah. Right now, we're trying to figure out if a single female can reproduce twice a year or not. That's our goal this year. We're not doing anything aging wise. But anyway, that's, if y'all want those fish, that's cool. And if you want to come out on the boat with us, that's cool too. We can only take maybe 2 of you at a time, because we've got a captain and another biologist, and because that work's going to start up in March, right? Y'all should tell us if you want to go out this year.
I believe we want to go.
Y'all are going to have to draw straws on who's going to go, because we're only taking 2.
There will be plenty of opportunities between us, between the agencies, and we'll just get you a signed volunteer form for the year.
We probably need to kind of be wrapping this up at this point.
Okay.
So if there's any burning issues we want to make sure we get raised and let the students think about, let's do that now. If not, I'm going to give you all haven't been shy about asking questions back, but is there anything you have on the tip of your tongues that you want to ask this assembled group? Going once, going twice?
I would say to you that writing any document as a team is really tough. Really tough.
We
So
don't
one of
know
the things
that.
I would recommend you doing is go back through your proposal and pull out the topic sentence of every paragraph, and then ask, do they make a logical string? Because it was very clear, different people wrote different parts and didn't always know what the other person had written. So it was almost like the different sections were themselves internally consistent, but weren't really merged together fully.
I think even when we were rereading it and had only like a couple of people going back over it, at that point we had looked at it so many times.
As far as the trees.
So I think waiting
Yeah.
a bit was,
Try giving it to somebody new that hasn't read it.
And one thing I want to say is, welcome to the academy, because you're not members of the academy until you do a proposal and it gets torn down and forced to limit the scope, far more limited than you ever would imagine. So, welcome, you're now a part of the team.
Bob, you have a question?
Trevor.
Sorry.
You had talked about the philosophical question that we should ask ourselves, even if there's an economic incentive, should we do it? I don't feel like that's really our job to answer that. That's DNR's job. We just provide.
No. Not in the proposal.
no, but think,
But that's your job as a member of society. It's your job as a scientist to make a determination of your sense of that values. You shouldn't turn off to the agency. They are entrusted by the citizens to manage, but they're only entrusted by the citizens. You have every right to form your own opinion as to whether or not you think an invasive species should be eradicated regardless of whether it's perceived by other members of society as a good or bad.
so you said you guys had different opinions. What do you guys think personally?
Well, and there's,
With respect to snakehead?
Yeah.
well,
I was just wondering.
from a whole, from an invasive species standpoint whole, obviously they're intrinsically bad. I mean, introduced species is AFS policy is that they're bad. It's not, I mean, granted, yeah.
Well, it's introduced and established.
Yeah,
Let's
we throw piranha in the river. Is it bad? It's going to eat some fish while it's there, but it's going to die. But that said, from an agency standpoint, I look at myself as a public servant, essentially. So if there was enough uproar, and we don't have the data one way, if we had data to show that snakeheads are decimating the system, which don't want to overstate in your proposal. We don't know that right now. We've done modeling work. It looks like they could impact large amount of bass, but right now, bass are doing okay. So
Bite your tongue, they are not.
So this kind of stuff is important. There comes a time with invasive species where you might just say, well, look, we can't control them. We're wasting our money. Let's put it somewhere we can. Lamprey was a great example. That's actually working. Asian carp, that's a huge example. They get billions of dollars to control Asian carp. We don't have that kind of money for snake. If we did, I'd say you wouldn't even be looking at snakehead research right now.
And that's
That's
exactly why we debated. I mean, Tom, I think that question is very important.
Mhm.
Even within the agency, we still debate whether the snakehead are invasive or not. Hell, among the agencies, right? We debate whether snakehead are invasive or not and whether they should be managed or not. Some state agencies think that we shouldn't be doing anything with them. We should just back off and not say word one about it. And some think we should go headlong. And then we've got some AFS groups, like the Aquatic Nuisance Species Panel, who really don't get that it's not an AFS group, the aquatic species, they don't really think that these introduced species are ever good. So it's a very philosophical argument. Mine is that, at least within I think that northern snakehead, they were recently introduced, there's probably something we can do about it, and if there's not, we can actually make people less fearful of touching the animal, which is where we were years ago, 2004, 2005, everyone got scared of them because it's a stupid snakehead terror movie, and everyone was talking about coming out of the water and eating the children. So as a management agency, our job was to make people less fearful and to encourage harvest.
Well, and right now, you're at that kind of crux, I think. Now it's been 10 years. They're still growing, they're still expanding. We don't know. They're probably not at an equilibrium, so we're kind of at that philosophical point where do we want to keep, is it worth pursuing, Can we control them? We haven't controlled the spread. Even if we can probably limit their populations through pressure, sure, but the spread, they're spreading throughout the Chesapeake Bay watershed, which is the larger concern, which is why a few years ago, we started shifting our focus to trying to understand more about them and the ways in which we could control them. So things like genetic population effective size and population structure within the Potomac could help inform management efforts.
But then we're going to get to that point, at which point do we stop investing that money, like with largemouth bass, and say, well, they're non-native, they're established, there's nothing we can do, let's just be happy with them.
Public attitudes are important, because right now there's probably a fair mix. Some people love fishing for them. We had a guy, he said, I was talking to him, he's writing a book on how to fish for them. And I said, have you eaten it? He goes, no, they're much too pretty to kill. So, and that said, I don't think anybody corrected you. It is, you can release a live snakehead if you catch it, but as soon as you catch it, you have to release. You can't transport it. If you decide to go back to the boat ramp, it has to be dead.
I think one of the things we talked about at our last meeting was how much is enough. Let's say 60% of anglers say we like catching them, and then 40% that we survey say we hate them, we want them out. Then we have to think about what we want to say and how we want to perceive that. So
I think that's a smart way to, and you should consider, like you considered lamprey, you should consider other invasives. But just remember, invasive is a subjective term, and there's a lot of societal influence on that. You can make the same argument for terrorism, right? It's a philosophical consideration. But there is no arguing that the species is introduced, and there's no arguing that it comes from a different continent. Now, whether it becomes invasive or not requires a lot of philosophical debate. And I think if you come to that point where you see that 40% of your anglers say, nah, get it out, 60% say, no, we want to keep it in, and by the way, we want to make it catch and release only, we want to keep all the big fish in the water, then you guys can philosophically decide whether that's what the best tactic is going forward. I mean, hell, we're having the same argument with blue catfish right now in the river. We're calling them invasive, but but Virginia doesn't want to, which is fine.
That's because they introduced them.
And some anglers want to keep the really big hogs in the water, which is legally they can definitely do. And so blue catfish almost become a more relevant example of what we're all talking about, whether it's an actual invasive or not. whether we should care about managing them or not, whether we should care about pulling them out of the water or
Lake
not.
Michigan is the perfect example for this, right? You talked about sea lamp prey in Lake Michigan, which you can control. They wiped out the native predators, which allowed an exotic alewife to invade. So they stocked salmon, Pacific salmon, to control the alewife. They were so successful, they generated this huge fisheries. They're now talking about having hatcheries to stock exotic alewife back into Lake Michigan to support the exotic predators that they stock in Lake Michigan. It's like a city pond that's 600 miles long.
Yeah.
We'll just stock it.
Yeah.
Right? It's a completely, and now zebra mussels have got in there, another exotic, and completely changed the food.
Just depends on who you ask, I guess.
Well, that's the fun part of your thesis to write.
Yeah.
And the flip side of the thing about it is, you know, you're asking people if they want to get paid, then you're actually creating that demand that is going to want to drive them to be around, you know, for the long term and not eradicate them. So.
All right. Well, I think we're gonna wrap it up. Again, I thank experts for clearly spending a lot of time thinking about this and reading your proposal and offering some fantastic.
Thank you.
Thank you very much.
They baited your hook.
No problem.
And
Do you have any
our library
questions?
staff, thank you very much for coming and participating in this, and we'll see how these the library award competitions shake out at the end. We got 11 more of these to go, right? So thank you very much for coming. You all need direction how to get out.
Nah,
Parking, that kind of
Pretty
thing.
good.
We'll figure it out.
Just in the nick of time.
Josh, can you send me that management report?
Yep.
Sure, good. God, it gets exciting.
And we only lost one other team. That's not bad.